next Wednesday at noon in this classroom. And most of the points will be from new material. And then in previous years, it's been something like 75% new material, 25% cumulative. But by Friday, I will give you the topics of the cumulative so that you know what to study specifically. So the way I picked that is I'm going through all the previous exam questions. And if I see something that I had really hoped that you would grasp the material and do a good job on, but that didn't happen, then I will formulate a new question about that on the final exams. For the cumulative portion, I tend not to do, I tend to zoom out, I guess. I don't get super detailed on the cumulative typically. I tend to zoom out and do a more general concept, but we can talk more about that on Friday, especially once you see the topics, then you can start asking me specific questions on that and I'll help you through that part. Okay, so I know that you're wrapping up the research projects as well. And so the presentations will be on Thursday in your respective lab periods. And if you have any questions on that between now and Thursday, feel free to ask me or Duncan, of course. And one thing that I'm not sure how much he emphasized or not on when you give your presentation, just make sure that you're pretty close to the time limit that's being asked of you. The reason that we do that is for you to get practice at adhering to a time limit. If you ever give a presentation somewhere at a conference or something, they put firm time limits on you. And if you go over, especially if you go over, they'll like yank you off the stage, even if you're in mid-sentence. They're like, yeah, you're done, get out. And so it's a good strategy, a good skill to learn. So please try to practice it enough times so we allow a little bit of wiggle room, but not a lot. So you can talk to Duncan more about that too. Okay. Okay, stress physiology. Plants get stressed out too. And so this is kind of where we left off before Thanksgiving break. We were looking at the variety of mechanisms they use to deal with pollutants. So the pollutants here are the red balls, and they're showing many different options here. So the below ground stuff going on with the roots, you have phytostabilization, which is basically a way to fix the pollutant in the soil system. Often they're showing that it goes from a circle to a square. So what they're trying to show there is that there's some kind of chemical transformation going on there in order to keep the pollutant kind of stabilized in the soil system and hopefully will minimize any damage. Phytostimulation is getting at the idea that often you're using microbes. You're stimulating the microbes to help break down the pollutant. And so they're trying to show that again by the breakdown of the red ball there. Then above ground, this is where you have to get a little bit more careful if you're trying to remediate a site, because now if things get up into the tissue, like phytoextraction, so they just extracted it from the soil, but they really haven't, the plant hasn't remediated it in any way, hasn't changed it. So that it stays as a circle here. So that means if an herbivore comes along and chews on it, it could create problems for the herbivore. Phytovolatilization depends on the chemical. Sometimes the chemical is changed before it's given out through the stomata and volatilized out through the stomata. Sometimes it's not. So you have to know your chemistry and the physiology of the plant to know if that chemical will actually get changed to less harmful things, or is it going off into the air just as harmful as it was in the soil. And then, of course, you have to worry about the ecology of the system there, what's going to be around and could get damaged by that. Phytodegradation is good. That's a really nice situation. They're showing that the ball is getting broken down. And so the idea there is that the plant will break down the pollutant to less harmful or completely harmless chemicals. And that's kind of the ideal situation, of course. When you consider plants, if you're asking, we've got this contaminated site, we want to try to grow some plants in there to phytoremediate it. How do we do that? How do we approach this idea to be most successful? Usually they'll pick a plant that's quite fast growing. Poplar, again, I think I might have mentioned that before break. Poplar trees or poplar saplings are usually often picked because they grow very quickly and they tend to have a pretty good root system. In general, you want rapid growth and lots of it. You want good biomass because that usually means they're able to really take up the pollutant and hopefully degrade it and have it in their biomass. Including the roots, you want good root biomass too. And obviously, you want the plant to be able to grow in that site. If it's not adapted to that site because it's too hot or too dry, then you have some issues. Definitely want to consider the root structure. And so you do have to know a little bit about the depth of the roots. You have to know a little bit about the architecture, surface area. You tend to want really high surface area because that allows for them to take up more from the soil, plus good surface area promotes lots of good beneficial microbes that are also hopefully remediating as well. Whoops. And so they've even been looking at aquatic plants for polluted aquatic systems, lakes and rivers. They've been looking at kind of putting out floating mats of plants where a lot of this research that I was seeing was done over in India in some of their polluted rivers. And they had really good biocontrol, not bioremediation with that effort, with the picking the right plant that had really good root systems. And then they also would inoculate bacteria in as well that were good at remediating. So it was a joint effort. So in general, you want a plant that has high transpiration rates, fast transpiration rates. And that usually means that they're taking up water in really nice rates of speed from the soil. So if the pollutant is dissolved in the water of the soil and they have that nice fast transpiration rate, that means they're able to get more of the pollutant out that much more quickly. You also want to look at what is coming out of the roots. And that's important to consider what impact that has on the microbes in the system. So root exudate, you want to think about encouraging coexisting microbes that hopefully are breaking down the pollutant. So you want well-fed, so to speak, well-fed microbes. And then you do need to know what is the plant doing with that pollutant. Are they sequestering it in their tissues? Are they breaking it down? Are they volatilizing it? And obviously, hopefully, they're thinking of what impact is that going to have on the ecology or the ecosystem? Are other animals going to be impacted? Not just humans. You have to think about the whole ecology of the system. And so there's a lot that goes into this. Definitely, if you can figure out that the plant does have the ability to break down the pollutant, that's like what you really want to do. You want to know the plant, yes, the plant can definitely break this down. Now, you have to think about what type of pollutant it is. And you have to do a little bit of digging into their genome, the plant's genome. Like, does it have the necessary genes to provide protein transporters, for example, so that the pollutant can actually be taken in and acted on by the plant? So what they have found is that most organic pollutants that are out there, they call them xenobiotic to plants. They're foreign to plants, which means plant didn't evolve in their presence. So the plant never evolved transporters to take those pollutants in. So that's a big problem. If they don't have a way to take in the pollutant, then they have minimal opportunities to break it down unless that pollutant has the mechanism through diffusion processes. It can get through the cell membrane. And that's dependent on the chemistry of the pollutant. If the pollutant has the right quality to get through that large hydrophobic section of the membrane, then fine, it'll come through via diffusion and hopefully the plant will act on it. But you have to consider the pollutant and if it has the right chemical properties to come across via diffusion. And some can and some can't. So many of the other pollutants that are non-organic, the plant has experienced those, like some of the heavy metals, for example, the plant has coexisted with those for long stretches of time. So they have the ability to, they have the transporters. So this figure, they're showing, for example, up here, they have the inorganics. And what they're trying to show with those gray boxes is that that's a transport protein that's allowing the inorganic to come in. You don't need to know the acronyms. That's not important, but basically they're showing that the inorganics do have these membrane transporters that are allowing the plant to pretty easily take it up and get it into their cells and tissues. And then they're showing here the idea that, one option is to sequester it so that the pollutant is very often they'll choose the vacuole. They'll stuff things into the vacuole because it's an enclosed organelle. And that organelle, it's kind of like a holding space for a lot of different chemicals. But if they put the pollutants in there, it's far less damaging to the cell than if the pollutant was just swimming around in the cytoplasm where it could start interacting and messing around with other things that are really important. So sequestering is a pretty common strategy. Down here, you're seeing organics, and they're not showing the gray box. They're just trying to imply that there's diffusion going on. And then they say, once the plant can get it in through diffusion, hopefully they can have some ability to break it down or conjugate it, kind of like the hormone idea. If you conjugate the hormone, it becomes inactive. Here they're showing that something is getting added to the pollutant. It's conjugated physically to the pollutant, and that can help stabilize it. And then sequestration, again, in the cell wall spaces. That's another place to stuff things for a plant. If they don't have an excretory system, they can't get rid of it like animals can when they get rid of things through their excretory system. So they tend to stuff it into the vacuole or into the cell wall space because that's relatively low damage in those areas. This is an abstract taken from a paper that some of the students in this class from years ago took. They did their research project and then they continued on with it for about a year and a half. And they were interested in looking at not just plants, but the mycorrhizal fungi that hang out with the plants. And if you have mycorrhizal fungi and the plant, can the plant survive in the presence of too much zinc? I mean, we need zinc to some, and plants need zinc, but too much of that is considered toxic. And so they were trying to see Lactuca is lettuce plants. So this is a greenhouse study that they did. And basically they purchased different mixtures of mycorrhizal fungi that you can get commercially. And then they grew the lettuce with and without the mycorrhizal fungi, with and without zinc present. And you can see where it says ZN, that's the presence of zinc. So these three were the treatments that had the zinc. You can see that the other three bars, there was no zinc added in, but the mycorrhizal fungi were added in. And so the BioAg and MycoBloom are two different commercial mixtures of mycorrhizal fungi. And you can see it without the mycorrhizal fungi, the treatment with just the zinc, you can see that the zinc concentration in the lettuce leaves was quite high. But you can see that you definitely see a decrease when you incorporate the mycorrhizal fungi. So this is implying that the mycorrhizal fungi are providing some kind of protection mechanism in the sense that there's less zinc making it into the leaf tissue with the presence of the mycorrhizal fungi. And then this lower graph, they're looking at amount of zinc left over in the soil at the end of the experiment. And again, if you don't have any mycorrhizal fungi, you can see you have a large amount in the soil, but there was again a reduction in the soil. So both of those taken together get you thinking that, well, it seems like the mycorrhizal fungi is taking up the zinc from the soil, but then sequestering it in their own tissues so that not as much is going into the actual leaves, which is important for people who then harvest the lettuce and eat it. You don't want too much zinc going into your body. And so being able to use the right mixtures of mycorrhizal fungi could also, help with this remediation effort, especially when you start thinking about food production. If you want to grow plants in iffy soil, which we have to do more and more of that because our human population keeps going up. We don't have as much land available. So what we do have is marginal, some of it contaminated. And so now they're saying, okay, how do we grow food? Can we grow food in contaminated soil? And so they're looking at mycorrhizal fungi as an option to kind of sequester and protect the plant. And this study is showing that it can do that. But you can still see that there's zinc in there. According to the company that we sent the tissue to have it analyzed, the level that was found in the leaves was safe to eat. So that's the other thing too, because you can have some zinc present, but you can't have, toxic amounts. So mycorrhizal fungi are being looked at as partners as well, not just bacteria that hang out with the plant. They're looking at the mycorrhizal fungi and its ability. They already knew that fungi can sequester certain compounds. That's why you have to be careful if you go forage for edible mushrooms. You have to be a little careful about that. You should know a little bit about the soil so that you're not getting toxic buildup in the tissue of the mushroom. Okay. So another thing to worry about with plants is the so-called salt accumulation. Salts are not just sodium chloride. It's a lot of different ions that are present in the soil system just naturally. And if you think about whether it's your potted houseplant or if you're growing indoors in soil, or if you are in an ag field that's been have heavy inputs of mineral fertilizer, those are salts as well. So the inorganic fertilizers that get put into the ag fields, that is the so-called salts as well. And what tends to happen is that especially in ag systems, you tend to, you do that watering system, irrigation system on the field. And then as the water is dehydrating out of the soil, just due to hot days, you start to get the salts kind of moving up the soil toward the surface due to capillary action. And what happens then is you tend to get a buildup of salts toward the upper layers of the soil where a lot of the roots are found. And so if you keep doing that over and over, and you can imagine fertilizing through the season and then the irrigation, if you're not careful, you can get too many salts accumulating right where the roots tend to love to be, toward the surface of the soil. So just taking one example, if you apply sylvite, that's an inorganic, it's potassium, and you have, all kinds of inorganic nitrogen and potassium and phosphorus. You have minerals coming in via precipitation and dust getting blown around. There's minerals in that too. So irrigation, you definitely don't have pure water that they irrigate with. So again, you have salts in pretty much any water system for irrigation of ag fields anyways. And so, the plants tend to take up the water. Sometimes they exclude out some of the salts because they won't need them. And if that happens over time, you can imagine all of this, including the evaporation of the water. You have that capillary action, you get a lot of salts accumulating. And this is a real problem in some of our agricultural fields. In fact, some have been completely abandoned, at least for a time, because they know that it's just too salty for the soil or for the plants to grow in that soil. And so they're actually actively looking for plants that can deal with salt stress and are trying to find, especially agricultural plants that have that ability. And they're starting to look at genetic modification techniques. And so they're turning their attention to CAM plants, because CAM plants, if you think back to CAM plants, they're the ones that live in deserts. And so they are adapted to really drought-prone areas. And in essence, drought-prone areas kind of have the same idea as really salty soil areas, because it creates a situation where the plant doesn't get access to water as it should. So if you think about it, a bunch of salts in the soil mixed in with the water in the soil, but you have just a lot of solutes. Well, that's going to drive your psi sub S really negative. That could actually create a situation where water comes out of the plant into the soil. So very similar to this idea of a drought prone area. So they're looking at, here, this is a study where they were showing the peptase production and amount of peptase protein in CAM plants that were grown in a salty condition. And you can see initially they didn't have much peptase up and running. Okay, these are facultative CAM plants, which means, remember, they can turn off the CAM pathway if they want to under good conditions. Well, you expose them to salt, that triggers their CAM pathway. And so peptase is the kickoff enzyme for that. And so you can see that it just keeps building in amount the longer that poor plant had to survive in that stressful salt condition. And so they're trying to understand, especially they're zeroing in on CAM plants and seeing if they can maybe create situations in other ag plants to simulate what's going on here and maybe genetically modify. So they're working on it, but basically what they see over and over, if the plant isn't well adapted to salty conditions, it's basically it's an osmotic stress. So the psi sub S component of the water potential equation is really impacted. And so what that means is your overall water potential then gets really negative, because we said before, more solutes, more negative psi sub S. And you can actually have water moving from less negative water potential in the plant to more negative water potential in the soil. And so it's actually sucking water out of the plant due to the laws of water potential and water movement based on that. And the other problem too, so not only is that can be happening, but then you get some of those salts, because they're in high abundance, they're, just too much of them, they start to mess with other solutes the plant needs, like the plant can't get to those. And so basically you get this competition between the salts and the needed mineral, and sometimes the salts win. And so the salt will come in instead. And so, for example, too much sodium in the soil system can make it really hard for the plant to get access to potassium. So it's kind of a double whammy where you have this water potential issue going on where you can actually get water coming out of the plant, plus you have this competition of the salts with some of the needed solutes, and then the plant can't get to those, can't bring them in. I think I may have showed you this diagram before where you can have a plant in water with good root system and you put it into water. So in figure B over here, that's, good water, it's pure water. Figure A, they dumped in some salts and you can see the plant starting to wilt and die, even though its roots are in water. But because you've added so much solutes, you've driven the water potential so negative. It's weird, but you can actually do that. And it's basically playing with the psi sub s of the water potential equation. And so, salt was added in figure A there so that it was creating an osmotic pressure. Your psi sub s was limited. It was too great for the plant to overcome. So even though its roots are in the water, it's just because it's so salty. It's really bizarre, but you can do that experimentally. And so it's actually creating a problem where the plant can't get access to free water. So this is going on in a lot of the soil systems that have been over-fertilized with inorganic fertilizers. Then you have drought-prone periods where you get a lot of evapotranspiration of the water out of the soil, drought, buildup of salts, and you're messing with the water potential, and the plant starts to lose water to the salty soil. So these are some images just trying to drive home the point that salty soil can be very damaging. So you get overall decrease in biomass. So they're showing the corn plant here, where the whole plant is basically much stunted in growth. And then you can see, of course, when you grow corn, you're going for the corn on the cob, and you can see how minimal the yield is and kind of deformed. And you can see in figure C, it's really bad where there doesn't even look like there's many kernels that got formed. So there's not much nutrition there at all. So this is definitely heavy chemistry. I'm not going to ask you to memorize any of this. The big picture concept that I want you to get from this is that if you have certain salts or minerals that are in excess in the soil, then it causes problems with the uptake of other key minerals. And again, this can get very complicated very quickly. And, if you ever go into a field of, a job where you have a particular plant, they tend to know, especially some very important food or medicinal plants, they tend to understand, which plants, which of these is more of a concern than others. But it's very complicated with the idea of, we know that there's all kinds of different minerals that the plant needs, but too much of anything can be bad, and it can start create these competition problems and cause this nutrient lockout where you can't get access, the plant can't get access to all of the necessary minerals that it needs. So this can happen in an agricultural field. It can happen in big indoor grow areas where you're working in a greenhouse and soil-based systems. It can happen to your houseplants that you have in your home or room. And so unfortunately, I've seen this happen way too many times where I'll notice my soil surface getting all kind of white and crusty looking. And I remember the very, very first time that happened, I thought it was a bunch of fungi because I'm very fungi centric. So I was thinking, oh no, there's an overgrowth of fungi. And so I was showing it to my one grad student, Scott. It was one of my work house plants that I have at work in my office. And I said, what do you think's going on here? And he came up to it and licked his finger. And then he licked or put that on the crust, and then he licked his finger again. I'm like, what are you doing? You shouldn't do that. And he's like, oh, it's just salt. So he was very confident it wasn't any fungi or anything nasty, but I still got after him for that. But yeah, it was salt. And so basically it's because I was growing in pots where you water from the base, which a lot of pots, there's a lot of them out there, but I did that for too long without flushing the soil or changing the soil, and the salt started to accumulate. So every once in a while, you should flush your soil with really clean water, even using like distilled water, and let it drain all the way out. Don't like keep the water in the bottom of your pot, just let it drain all the way out. And that will help get rid of the buildup. You will notice that not only will you see the top of your soil getting these weird crusty white and yellow growths on them, but it's just the salts precipitating out. There's so much in there that it's precipitated. Then your plant will start to suffer shortly thereafter. You tend to see lots of browning along the margin of the leaves and the tips, and that's probably because they're sequestering the toxic amounts, the excess amounts in those areas of the leaf. And so you can see that starting to show up as the symptoms. So you can even see it like this is a clay pot. I've seen that before too, where the outside surface of the clay pot gets all salty looking, like caked. I don't advise licking it to determine that, but chances are this is a close-up of the top of the soil. So you can see the buildup. So once it gets like this is pretty bad. And so often what I do, if I start to see this, I will excavate that top layer of soil out of my pot, plus do a flush, plus retop it off with better soil. Either that or just transplant the whole thing into a brand new pot of soil. That's probably the best, but often I don't have time to do that. So I do the top portion. So just keep in mind, it does matter a little bit how you water, how quickly you'll see accumulation. So if you water from the bottom, you tend to, especially in pots, you tend to see build up a little bit more quickly. But anyways, the basic idea is, especially in a potted plant, I mean, that's such a small amount of soil. It's pretty easy to overdo it if you over fertilize, and you can really stress your plant out. So make sure that whenever I fertilize my plants with an inorganic, I look at the directions on the bottle and then I cut it in half just to try to minimize the salt buildup issue. So here are some symptoms where you can see the browning along the edge and tip and just looking really sad. This one, of course, and all the necrosis. So you've basically killed off. I have one in my office right now that's doing that. And so this idea of too much of the salts, you often hear the term nutrient lockout. That's basically saying that it's messing with the ability of the plant to take up key nutrients because there's too much of the other salt. And you can also exacerbate this problem. You can make it worse if the pH of your soil is off. And so that's going back to this idea. You've seen this already when we looked at mineral nutrition, how you have generally they say a pH of 5.5 to 6.5. That's what you want. If you tend to get, if you look up into the alkaline condition, like 7.5, you can see manganese really suffers, phosphorus really suffers. You don't get much access to that under alkaline conditions, for example. Or if you go the other way too. So you can, if you don't have a good pH of your soil system, you definitely can cause additional problems to getting these deficiencies. So both of those are really important, especially in potted plants. So this figure is just trying to show how impactful this problem can be. And so I know a lot of people are like, oh, salt, what's the big deal? But actually, salt is more than sodium chloride, although plants really don't like a lot of sodium. Sodium is not good for plants, just like it's not great for us either in higher amounts. But it's not just sodium chloride. There's all these other salts, these minerals, these ions that are in the soil. So you can see here, we talked about, they just have a hard time taking up water. You have nutrient lockout often where key minerals can't get in as easily. You have for legume plants and other plants that do nitrogen fixation with their bacterial partner, that can really suffer as well. So that's a bad situation. And then it just impacts the plant negatively. Salty soil, the root systems aren't normal and they're decreased. They're not as, surface area isn't as good. So there's a lot of impacts both on the morphology and physiology of the plant and the associated microbes. Too salty for the plant usually means too salty for the microbes in the soil too, and they have a hard time. even to the point where they're showing, damage to the DNA. So that's like the ultimate. So big problem. And so many of our agricultural systems, our soils, like I said, they've been over fertilized over the years. So this has accumulated over the decades and it's created a lot of ag fields that have to be abandoned or remediated in some way so that we can get back to using those fields where we can get good biomass from those plants. So if you have potted plants, keep your pH in the good range. It depends too on your plant. Some plants have slightly different pH ranges, but mostly you can assume 5.5 to 6.5. And then just be a little bit aware of if you add in peat moss is very acidic. So just keep an eye on that. You'll might need to think about that if you put in a lot of peat moss. Don't over fertilize on a regular basis. And the mineral balance is hard. It's hard because the way to detect individual minerals is we don't really have the technology for that. You can buy a probe that measures electrical conductivity. We have one of those here, but that gives you an overall value, which is useful, but it doesn't tell you if you're low in one nutrient over another. So it's an overall value, but it can't pinpoint exactly which ion might be low or too high. So that's a problem. It's a good idea to either repot your plant into fresh soil on a regular basis or flush with some nice clean water on a regular basis. And then this is the same simple trick to get rid of chlorine. If you have a watering container, just fill it and let it sit for 24 to 48 hours. And that will dechlorinate. The chlorine is a gas, so it will come out of your tap water just by letting it sit open to air. And so I do that with we try to do that in the greenhouse as well to degas it. And so that's a simple thing to get rid of the chlorine. So hopefully the technology will get a little bit better, but right now it's expensive. They're trying to come up with a less expensive tool that can be sold for pretty much anyone to use. But to do ion selective electrode testing, I mean, you can do it at a university, but most people don't have access to a university. And so when I was looking at what's out there currently, this is a paper from 2018 and they're making progress on it, but not quite there yet. And hopefully, like I said, hopefully that technology will get there. But right now, the best you can do is electrical conductivity, and it just gives you an overall value. So, but they're working on it. But right now, it's very, very expensive if you want to do that. And I just wanted to bring up the point that we've been talking a lot about if you think about remediation, we think about, plants being able to remediate the associated microbes in the soil. Don't forget about the endophytes because they are also being shown to be good remediators. And so here's titles of 3 different published papers where they're looking at the use of endophytes for really salty soil or droughty soil. And so again, this idea that the endophytes help the plant adapt to these conditions. And so it's really the endophytes that are driving the ability of the plant to grow and have a successful life cycle there. And so they're really looking into that too as a way to partner the right types of endophytes with the right qualities to help these plants deal with growing in these salty conditions. So definitely making some progress on that, but again, there's a lot of work to do, especially when you think about salt stress. There's a lot of that going on in agricultural fields, unfortunately. And hydroponic systems, you also have to be really careful about that. Okay, so I'm going to stop there with pollutants and salt stress. I did ask or before break, I asked a question out on the EduCat. What other stress topic would you like to think about? And more people voted for chilling and freezing. So I wanted to talk a little bit about that. Let me get that going on. I don't know that that's in EduCat. Okay. Well, now it's there. Okay, some of the front slides you've seen already because they were in the other slide set I just went through. So I'm going to skip through those. Okay, so this slide with chilling and freezing. So chilling and freezing, which we have going on now in this area, beautiful snow and freezing temps. So chilling and freezing means something different in the plant physiology world. When you say there's chilling conditions, that means it's not freezing yet. The plant is not showing any frozen tissue, but it's not growing either. So it's suffering. It's not able to grow, but there's no tissue damage yet. And so chilling means something different. You can get bouts of plants getting chilled, but they survive it. So if you think about some of the southern areas, southern states, I'm sure you've heard about southern crops going through chilling, unexpected cold temperatures with, during the orange growing season or lemons or limes when you have all of that going on. But they can often make it through that. Not always, but often. And so chilling is more of an issue when you think about tropical or subtropical areas that have an unusual weather pattern come through where you get this rare chilling event that, hopefully the plants have adapted to it. When you're looking at this figure, what they're trying to show is two different types of plants. So the black balls are just kind of normal plants that are grown at normal altitudes. And then they're showing in the red here seeds that were from plants that were grown in high altitudes, which generally means cooler conditions on average compared to, ground level, base level atmosphere. And so the higher altitudes, those plants in general are just better able to handle chilling. And so this idea, there's a big push, in fact, right now in the UP and in the Marquette area. There's a lot of entities that are very interested in having like experimental fields. I know of a couple of groups that are doing this. They have fields where they're growing crops into really as far as they can into fall to get them to form seeds and then harvest the seeds and use them again here. And so the idea is if you buy seeds from some seed company, where were those plants that produce those seeds growing? Were they in a lovely greenhouse where they had perfect conditions, never were challenged with chilling or freezing conditions? well, then those seeds might not work so well up here, where we have a very short growing season. We often have chilling conditions, even in winter. I remember one 4th of July, I was wearing a parka because it was snowing on the 4th of July. So we can have a lot of those events in this part of the world. And so the idea of buying seeds from a plant that was grown under perfect conditions and then plopping them in the soil here probably is setting them up for failure. And so the idea is, let's grow local, get those plants acclimated to this weather system. And then when they form seeds, save the seeds and again, give them to the people in this area to grow. And so we're trying to create our own seed bank of seeds that have come from plants that can be acclimated and can tolerate some of these conditions that we're faced with here, such as chilling and freezing, even in a normal summer up here. So this idea, I think if you end up growing plants at any point in your life, especially if you want to eat those plants, you should give that some thought of where are you buying your seeds compared to where are you living. It can make a big difference. If you look at chilling impacts, when they do controlled studies where they make the plant go through chilling conditions, what they tend to see is photosynthesis definitely takes a hit. You get that slowing down quite a bit, which of course means not as much ATP and sugar gets made. And so the overall biomass of the plant will definitely get a hit. The other thing they see is that sugar translocation gets diminished quite a bit. And so, I mean, photosynthesis and phloem translocation to get the sugars to where they're needed through the plant. I mean, these are two huge concepts in terms of what the plant has to do on a day-to-day basis. The other idea is that their respiration slows down. So the respiration, we haven't really talked about that in this class, but remember, respiration is just the idea of the plant using oxygen to get their ATP, you know, just like what you're doing right now. And so that gets lowered. Their protein synthesis slows way down. And another big hit is their cell membrane integrity can get damaged. And so cell membranes, they're supposed to be semi-fluid. If you start to chill membrane material, it kind of gels up and gets too thick, so to speak. And then you have transport processes getting affected dramatically by that. So I think I'm going to stop there. We'll get into that idea a little bit more, but it'll take too long to do it in 1 minute. So we're going to stop and we'll continue on with that on Wednesday. Okay.